My today topic is a multiplex PCR assay for identification of major mastitis causing pathogens in buffalo raw milk and their evaluation of sensitivity. My name is Dr. Mohammed Safdar. I am lecturer in Department of Breeding and Genetics at Cholistan University of Bahawalpur. Overview, introduction and need of the study, objectives of the study, material and method, results, conclusion, and finally there will be references. Introduction and need of study. Buffalo milk is a white and shade liquid to nourish the young ones. It is easily digested and it is used to reduce the calcium deficiency in old and young ones. It is necessary nutrients that are present in the milk to boost the immune system to mature the young ones. Buffalo milk is changed into a number of dairy products such as ice cream, cheese, butter, yogurt. So, milk is pure from any contamination in udders, while in case of mastitis. What is mastitis? Mastitis is an inflammation of the udder and is common in dairy herds, causing important economic losses. So it cannot be eradicated, but it can be reduced to low level by good management of dairy animals, as it shows in the figures. The most common pathogens are Staphylococcus aureus, Staphylococcus agalactiae, Dysgalactiae, Baris, E. coli, etc. that can cause occasional heart outbreaks. So these figures are available here. The course of this infection varies. Most commonly, it persists for weeks or months in mild form, which is not detected by stockmen. The objective of this research was to develop a sensitive, specific, and economic multiplex PCR assay for identification of these major mastitis causing pathogens, and then to evaluate different antibiotics on major mastitis pathogens to check their susceptibility of drug-resistant bacteria, which were isolated from raw buffalo milk, as shown in this figure. Material and method. Here we can see we have cultured different buffalo milk for identification of pathogenic bacteria. And then we isolated those cultured bacteria and also we have identified them. Then we have extracted DNA from these bacteria and also we have designed specific primers of these species and we have identified them with the help of PCR. Where and how we have collected samples. 200 samples were collected from the different areas of district Multan, such as Multan Sadar, Multan City, Jalalpur, Pirwala, Shujabad. Samples were collected in Falcon tubes from January 2022 to February 2024. Immediately, they were transported to genetics lab and stored at -20. Milk samples. After that, milk samples were diluted and poured on various agars media for culturing. And then we have collected for DNA extraction. And finally, we have optimized simplex and multiplex PCR. The results shows that in the first here we have seen there is a ladder. It has been used as a control, then NC1 and NC2 were used as a negative control. And then sample 1 is specific for bacteria, then sample 2 for specific another, 3, 4, 5. And finally, we have optimized multiplex PCR 1 and multiplex PCR 2 Multiplex PCR 1 and 2 are only repeated one. We don't have made any change in them. Then we have checked the sensitivity of this hexaplex PCR for targeted bacteria. And we have started in the first sample 50 nanogram DNA, then 25, 15, and we have found 0.01 nanogram in one tubes. This was our limit of the hexaplex PCR. And it shows a very sensitive hexaplex PCR as compared to others we have done a thorough literature. Then we have calculated percentages of contaminated samples with different pathogenic bacteria at Multan district, and E. coli were 35%, SRS 25%, pneumonia 15%, and others were 10, 8, and 7% bacteria. And then we have checked their sensitivity on agar plates with the help of disk diffusion methods. And they shows A and C shows they were sensitive to bacteria, while B and D were partially sensitive, while F, G, and H were 100% resistant to E. coli bacteria. Conclusion. Specificity of PCR shows that reference samples were 100% accurate. They were identified with the help of this multiplex PCR. Sensitivity results for reference samples indicated that sensitivity of this PCR assay was up to 0.01 nanogram. By using this multiplex PCR, the isolates were identified, and out of 200 samples collected from different areas of district Bahawalpur, results had shown the presence of different bacteria with the percentages as they have been shown in this slide. The results suggested that the developed multiplex PCR was sensitive and specific for simultaneous detection of all possible Mastitis causing pathogens that has been collected from raw buffalo milk. These are some reference. I would like to thank you, all of you.